bad joke, and that climate change is impacting our coastal ecosystems, mainly by sea level rise. So mangroves are expected to respond to sea level rise by encroaching landward, and they're encroaching into different habitats. So salt marsh is the focus of this talk, but pine uplands are also being lost as well. So management, people involved in management are interested in mangrove encroachment, not just here in Florida, but across the globe. But not all boundaries are created equal. So again, I told you mangroves are encroaching into different habitat types, and the structures of these boundaries are very different. One function of boundaries is their permeability to mangroves, and so that's one of the focuses this talk is, why is it important to understand the permeability of these landward boundaries of mangrove habitats? And that is because the landward dispersal of mangrove propagules is this first step in mangrove recruitment. So mangroves can establish themselves landward of their existing boundary unless they can first get their seeds there. So this is Abyssinia germinans, the black mangrove. This is the species that I work with most often because it is found at the landward boundary of mangroves. Most of us are familiar with the red mangroves with the gigantic properties, but that won't be any part of this talk today. So this is what our mature mangrove looks like. You can see below it, there's some salt marsh plants, and so obviously these plants are going to be impacting the dispersal of the mangrove seeds. But those aren't the only factor. It isn't the only factor that controls dispersal. The shape and size and buoyancy of the mangrove propagules can also influence their dispersal patterns. So I just wanted to show you, this is what the propagules of the black mangrove look like. Again, they're very different from the red mangrove, so we would expect them to disperse differently. And the mode of dispersal is through hydrology. So basically these propagules are buoyant. They move when tides are present or rain is present to move them around. So the question I asked in my research was, how do propagules move across these landward boundaries that are already occupied by salt marsh plants? So the purpose of our experiments was to determine if the plants that occupy this boundary affect propagule movement. And the first step was to see, okay, how well do these salt marsh plants entrap mangrove propagules? Can they physically stop them from moving? And then secondly, we wanted to see, okay, the propagules that are moving, how far in which direction are they moving in? Our study site wasn't actually in Cyprus, it was nearby. It was within the Mercury Bay National Estuary Research Reserve on Cannon Island, which is just across from Key Waken. This is what it looks like when it's nice and high and dry out there. You have the mangrove lined tidal creek in the back of the island. At the interior of the island is a salt marsh, and then at the other side of the island we have Australian pines. Most of the year the tides come up to where the established mangrove tree line is, but during the fall we get some really extreme spring tides, and this does bring in tides that are sufficient to inundate the entire island. And this is the only chance that the propagules get to disperse in this landward direction and colonize these interior areas. So this is when we set up our experiments. During these high tides, the propagules are interacting with several different salt marsh plants. These are the most common plants that we have on Cannon Island. We have grass, Spiralis virginicus, and two other succulent plants. That is Maritima and Susubium portulicastrum. Don't worry, I won't use that the rest of the time. We've got a grass and two succulent plants. So our first objective was to see how well these salt marsh plants with different growth forms trap propagules. And by entrapment, we basically just mean how many propagules were still where we put them over time. We looked at the percentage of propagules that were within our experimental plots. We completed this in 2010, and it is published in Ecology if you want to read all of the details. But basically, we did find that salt marsh plants in different growth forms differentially entrap mangrove propagules. The basic experimental design was to weed salt marsh in the middle of August during the fruit fall period and to establish a system of treatment. So we had a natural polyculture that contained the grass and also at least one of the succulent plants. We also had monocultures of the grass and of both of the succulent plants. So we had natural polycultures, a grass monoculture, a monoculture of the succulent Batis, and a monoculture of Susubium. And we replicated these in the marsh. We emplaced propagules into these plots, and we looked at how many were still there over time. And in order to understand how the salt marsh plants were influencing the entrapment, what we did was take these digital pictures of the plots, and when we digitized them, we were able to make them into binary images, which is what you see at the top. And we were able to calculate the number of pixels that was vegetation versus background. So this was kind of a quantitative measure of vegetation structure, which is the next point. When we looked at how well plots and tract propagules relative to this measure, we found a really tight relationship. So the plots with the grass, the natural polycultures, which are in black, and then the grass, which are in red diamonds, those both tract propagules significantly better than either one of the succulent treatments. So building on this first study, what we did was we executed a second field study in 2011 to determine where the propagules are moving and how far they're moving if they're not tracked. And so this is our general site layout. I'm just going to walk you through the experimental design because it's not super intuitive. We do have a tidal creek at the back of the island. The mangrove fringe is along this creek, and then the salt marsh plains are at the interior of the island. So this is our land to sea gradient. From now on, everything at the bottom of the screen will always be seaward. But it's not quite this simple. These are our black mangroves. The bigger ones are mature mangroves. The smaller ones show small recruits and seedlings. So you can see there are some mangroves within the salt marsh area. But it's not quite so simple. It's not just one type of salt marsh. Again, there are succulents and grass, and there's the zonation. So the succulent plants tend to be lower, near to the mangrove margin, and the grass dominates the upper interior of the island. So we're really focused on this middle portion where the succulent plants overlap with the grass, because this allows us to do manipulative studies at the same tidal height. So the experimental design was to establish plots in this area where the plants overlap, manipulate the plants within the plots, put in some marked propagules. We used spray paint because it's been shown not to influence the buoyancy of these things, and it's kind of fun to track how pink and orange propagules. And we followed the dispersal and used statistics to analyze the patterns and see if they were different than chance. So again, this is our salt marsh layout. The first step in the experimental design was to delineate what I'm calling the conspicuous interface between these two salt marsh plants. So basically, if you go out to the marsh, it's very obvious where the succulent plants were dominant and where the grass was dominant. It was a very constricted boundary between these two. So we just kind of eyeballed it. We call that 0 meters. And for the rest of the talk, if I say seaward or landward, it's always relative to the 0 meter position where the succulents transition to the grass. And so this is where we put our plots. We had 10 plots in total. There was a landward and a seaward part of each experimental plot. And so then we weeded these things because it's fun to weed plants in the middle of the summer. And so we had some plots that we weeded so that they only contained the grass in the seaward and landward portions of the plots. And then we wanted to exaggerate the natural boundary in the second treatment. So basically, we wanted to remove all the succulents that happen to be spread with landward, and then we also removed the grass that was present seaward of this boundary, so we had a very clear boundary in these plots. So this is what it looked like out in the field. First, we removed the succulents from the landward portions of the plots, and then from half the plots, we removed the succulents, and the other half, we removed the grass. So these are our two treatments. We've got the Sporulus virginicus only, or the grass treatment, and then we've got the succulent grass boundary treatment. And so this is what it looked like. I spaced them throughout the marsh. So there was, tidal creek. Some areas might have higher flow than others. So it's just I counted for that. This is what it looked like in the field. This is our lovely mangrove margin, and this is again our 0 meter position. So we put propagules, again bright orange in the grass plots and bright pink into the succulent grass plots, right at this 0 meter position, so in a line. And then we followed these guys. So this is what it looked like actually in the marsh. We've got these hot pink and orange propagules lined up, ready to go. This was done at low tide, and then we followed them after high tide events. This is our 0 meter position, and this is again just to orient you as to what I've been showing you on the cartoons. We wanted to know which direction are these propagules moving in, and how far do they go. We expected that all the propagules would tend to move seaward, that they would get picked up by the tides and carried out. That's kind of the dogma that exists. But based on our experiment from the first year, we thought since the grass traps these propagules and hinders their movement, we would expect them to move maybe just a little bit seaward compared to those that were obstructed by the succulent plants, which didn't entrap propagules very well. We expected these would move much further seaward and that we might actually lose some of these into the tidal creek. We didn't know what to expect in the landward direction. There's very, very few studies that examine the landward dispersal of mangrove propagules, but we didn't expect to see a difference in treatment because both plot types just had grass in the landward portion. So what did we find? How did these boundary types affect propagule movement? Well, we found that propagules moved seaward and landward, and that was pretty much true in every plot that we had. So each one of these dots is an individual propagule of where we found them the first day after we put them out. This was about 2 high tides later. But overall, we found that most of the propagules moved seaward, so that's our big blue chunk of this circle. We did have a really high recovery of the 240 fruit we put out. We found 233 in that, so that was pretty good. For those that say no movement, you can see that's a pretty big proportion of the propagules that we found. Those were within 5 centimeters landward or seaward, so negligible movement in either direction. When we break this down by treatment, we can see that there was a significant effect of treatment on propagule movement. The propagules that we put into the treatment that only had succulent plants obstructing the movement did have a higher proportion of propagules moving in that seaward direction relative to the propagules that were surrounded by grass and weren't able to move very much. And so if you're interested, this was a repeated measure, so and it was significant. So again, we've got some statistics up here too, but basically it just means that the orange bars are larger than the pink bars. We wanted to see what the overall mean distance moved was. So for every propagule, if it moved seaward, we tally that as a negative value. If it moved landward, we tally that as a positive value. And again, overwhelmingly the trend was for these guys to move seaward. So all of these are in the seaward direction. So the propagules in the succulent treatment, or the succulent grass boundary treatment, did move slightly further, and it was significant, and it was in the seaward direction. We stopped sampling after about 6 weeks in November, and that is because at this time about half the fruit that we put into the plots had rooted, so they're no longer moving around anymore. And 25% of the ones we emplaced were not only rooted, but had actually developed their first leaves. They weren't dependent on the cotyledons anymore. And you can see that the hot orange spray paint didn't seem to affect their growth at all, and there wasn't an effective treatment on establishment. So basically we found that salt marsh grass retains propagules very well, that a greater proportion of propagules moved seaward if they were only obstructed by succulent plants, and that propagules obstructed by succulent plants dispersed further seaward than those obstructed by grass. And so we're confirming findings by others that have seen that generally propagules tend to be moved seaward, but this study is novel because we looked at the landward dispersal of propagules, and we did find that some of the fruit that dispersed landward did establish there. And this is important because it's not just here in Florida that we have this Abyssinia spiralis, or this black mangrove salt marsh grass ecotone or boundary. We have very similar systems worldwide. This is a picture from Cannon Island. You can see our mangroves and salt marsh. And there's the exact same species, well, same species of salt marsh grass, same genus of mangrove in Australia. So we expect that these boundaries are going to work very similarly to what we've looked at in our system. So basically, when you're considering how mangroves are dispersing landward, how that first stage in recruitment, it's not only important to consider the biological factors like the plants that are occupying that space, but also the type of mangrove that you're interested in. Obviously, our black mangroves and the red mangrove propagules are very different, and we would expect the patterns to be very different for those two based on their characteristics. And then also the abiotic factors of sites should be considered because the tidal patterns and the precipitation regime are going to be very different. These can definitely influence the dispersal. And finally, salinity, especially if you're looking at dispersal of an estuary gradient, would be very important because it can affect the longevity and buoyancy of these propagules. So I'd like to thank everyone that funded my research and those that helped execute the labor, skill work, Beverly and Jill in the back from the Recruiting National Estuary Research Reserve, and of course the University